Welcome to Single-Nucleotide Polymorphism and Mutation Analysis and its Impact on Personalized Medicine. I'm Timlin Oliver, Managing Editor of GEN, and I'll be the moderator for this webinar. As a result of single-nucleotide polymorphisms, proteins encoded by affected gene sequences may exhibit functionally relevant amino acid substitutions, post-translational modifications, and structural changes. With the advent of personalized medicine, characterization of structural and functional changes in proteins that cause disease has assumed increased importance, both diagnostically and to determine individual patient responses and reactions. Our two speakers today, Pierre Thibault, Principal Investigator, Proteomics and Bioanalytical Mass Spectrometry Research Unit at IRIC, University of Montreal, and Paul Thomas, Research Assistant Professor, Department of Molecular Biosciences and Assistant Director, Proteomics Center of Excellence at Northwestern University, will provide different perspectives on the study of protein variants. Paul will examine the role of top-down proteomics, and Pierre will talk about Mhc Peptide immunotherapy. Our speakers will share their experiences and insights with us in just a minute, but before we get started, I want to encourage you to submit questions for our panelists. We'll answer as many as we can after the final presentation. Paul, you're up first, and we're ready to start.
All right, thanks. Good morning from Evanston, Illinois, and thank you for giving me the opportunity to speak today. My name is Paul Thomas, and I'm the Associate Director of the Proteomics Center of Excellence here at Northwestern University. Today, my talk will be about understanding biological complexity with top-down proteomics. And we feel that top-down proteomics is well-suited for understanding these problems, and certainly as they apply to personalized medicine, and as it avoids artificial complexity that can be generated through proteolytic digestions. So the central dogma of biology states that DNA will encode for RNA, which will then encode for particular proteins. And one thing to take away from this basic fact of biology is that at each step, a variety of complexity can be introduced. DNA can possess different coding polymorphisms. RNA can be alternatively spliced into a variety of different forms. And proteins themselves can be covalently modified. So in higher proteomes, we look at these changes in two separate ways. The first are large changes to a protein sequence, and we call these isoforms, different isoforms that are made. And these come from RNA splicing, so alternative splice forms, as well as endogenous protein cleavages, and alternative promoter usage, which may change the base sequence of the form that is observed in nature. So further adding complexity here is what we term the proteoform. And the proteoforms are the complete set of modifications that can occur on an amino acid sequence. And these modifications include known post-translational modifications, acetylation, methylation, phosphorylation, single nucleotide polymorphisms that change just one amino acid, and any unknown or unexpected modifications. To understand the large number of protein forms that can be generated from a single gene, we developed this method known as shotgun annotation, in which we can go through the process of the central dogma from a gene to RNA and into proteins and observe how many different protein forms can be made. So in this simple case, we have one host gene, which has an alternative, as a SNP present in the red exon. This can be constitutively put together through splicing in two different ways, generating from the one host gene, which had two forms, into four now mRNAs, which are translated into proteins. And with the addition of even one post-translational modification, we see that from one gene, we've already produced a potential of 8 different proteoforms. So the technical hypothesis that we forward here is that the measurement of intact proteins, or isoforms and proteoforms, will provide a stronger correlation between mass spectrometric data and complex biological phenotypes. As a bit of introduction of what top-down proteomics is, we'll examine its difference with bottom-up. Both start with an extraction of cellular proteins. These proteins are in bottom-up, either separated and then digested, or digested and then separated. And then the peptides themselves are analyzed with MSMS and are put back together in a variety of different bioinformatic ways into one protein. In top-down proteomics, we remove the digestion step and instead just fraction the intact proteins and perform MSMS on the intact protein forms themselves. We then fragment those, and we put the fragments back together into the proteoform that was identified. This has many advantages that we'll see in the future. One such advantage is that covalent modifications, such as post-translational modifications, may not be captured in a typical bottom-up experiment, but because a top-down experiment includes fragmenting the entire form, the post-translational modification should appear within the fragments that are generated. So to develop a general platform for the identification and characterization of intact modified proteins, there are several steps which we must go through. First is a separation. Then we measure the intact proteins, we fragment them, and then we put them back together. We have the front-end separation, instrumentation, and then the back end. So the top-down proteomics workflow, as we recently published in Nature, for high throughput consists of two separate separation steps, one by PI and isoelectric focusing, and the second by molecular weight with the gel-free process. Samples are cleaned up to remove the SDS present in gel-free. The samples are then analyzed with nano-LC FT-MSMS, in this case on a 12 Tesla LTQ FTMS, and the data are then processed with ProCyte PC. So gel-eluded liquid fraction entrapment electrophoresis was developed by a gentleman named John Tran, who is a postdoctoral associate in our research lab. And the basic concept is that of an SDS page gel, where in this case, instead of having a slab, you have a column of SDS page resin. Proteins are loaded onto the head of the column and are separated by molecular weight. So the smallest proteins elute out first into a collection chamber filled with running buffer. These proteins are then removed, and new loading buffer is added such that the rest of the proteins can be eluted. Now, if you take a small portion of these fractions and run them out on a typical slab gel, you can see the typical separations that we can achieve in this methodology. For top-down proteomics, there's two major requirements of instrumentation to make this happen. The first is a high-resolution mass spectrometer for both intact and fragmentation data. And the second, somewhat related, is low part per million mass accuracy such that we can have high confidence in the results that we obtain. The reason high resolution is important is that as we move from a small molecule world to a peptide world to a protein world, we see that the number of isotopic peaks that are generated when one goes to even a small protein are much larger and much more tightly spaced than that in a small molecule or a small peptide. So because of that, we need a high-resolution mass spectrometer to really separate these different isotopic forms of large proteins. So FTMS meets that goal, and we typically see resolution resolving powers from FTMS of 1 million or 100,000, which allows us to not only see the charge states, as you can see in the top of this slide, but also the different isotopes rising from carbon-13 that are present in the forms. And we get this at a mass accuracy of 10 part per million or better. So the typical steps for a top-down identification is that a protein is identified in its intact form. It is fragmented through any one of many fragmentation techniques. And then ions typically contain either the N or the C terminus of the protein. And from that, we can put together a map of the fragment ions that were identified. And this is an example from 1995. So top-10 proteomics has been around for quite a while at this point. and is now really coming to its own in high throughput. We'll revisit this slide for top-down proteomics and high throughput, where we've replaced the manual step of infusion of an individual protein into the mass spectrometer, and we replace that instead with an online NanoLC FDMS-MS device. So protein separation with NanoLC FDMS-MS is actually quite simple. We do this on a typical NanoLC column. Proteins are then eluded out over time, and you can see that in this case, this is one gel-free fraction containing proteins between approximately 13 to 18 kilodaltons, and each of these distributions are separate intact proteins which are identified at different points in time through the chromatogram. And so if one was to zoom in on one of these charge states, you can see that the charge states that we see for the 17,279 species are actually made up of these isotopic peaks as well. And more importantly, the chromatography is actually quite good for the same protein. It elutes within 30 seconds to 60 seconds. And while that's not the case for all proteins, in certain cases it works out quite well. And so in this one chromatogram, we were able to detect 45 different proteins. So to detect these proteins, we go about a different step than typical bottom-up proteomics. In this case, we use the UniProt entry for Homo sapiens, of which there are approximately 20,000 that have been manually curated. In our database, we capture all of the complexity, or at least as much as we can, that has been ever seen on these proteins before and is cataloged within UniProt. These include peptide cleavage events, known modifications, sequence variants, and the like. We reduce the redundancies that can happen from different gene forms producing the same translated protein. And then we allow up to 13 separate post-translational modifications per sequence. And this generates approximately 50,000 unique base sequences and 8.5 million unique proteoforms, occupying about 5 gigabytes in our typical human database. So I wanted to finish here with several different examples of biological complexity and where top-down proteomics can really shine. So in 2006 or 2007, our group published a paper in which we were able to, so we took samples from the blood bank, isolated white blood cells from them, and performed top-down proteomics. And in 3 of the donors, we noticed that the Charcot-Leyden crystal protein, we were able to see that these subjects were heterozygous, possessing both an alanine and a valine for a difference of 14 daltons between the 3 forms. As well, we found one individual with a difference in acyl-CoA binding protein, the valine being swapped for methionine, and all of these changes were verifiable by PCR. But in my opinion, top-down proteomics is a very expensive PCR process. So there are better ways to use mass spectrometry to determine changes in biology. And I'll go through a couple of them from here. The first is I'll share with you a study from a group at Merck Proteomics, which at one time was the largest investment in proteomics in pharma. And they published this paper in 2010 on the quantitative analysis of intact apolipoproteins in human HDL by top-down differential mass spectrometry. And the main question that wanted to be answered by top-down was, can a specific proteoform be associated with a specific biological phenotype? And so in this case, there were 6 subjects with the only major difference between them in their HDLC, seen in this third column from the right, where the first group, the first three, had a lower HDL than the second group. The plasma from these, or the HDL fraction of the plasma from these patients was then separated, and proteins were identified. And so one protein that was of particular interest showed a drastic difference in its level in the low HDL group versus the high HDL group. And it turns out that this protein was isolated, fragmented with electron transfer dissociation, analyzed with ProCyte PC, and identified quite conclusively to be apolipoprotein C3, with a post-translational modification of a triglyceride on this threonine near the C-terminus. So given this lead, the researchers wanted to look at the different proteoforms that were present of this protein. So there were 3 proteoforms identified. The first is identified by circles in the upper right-hand plot and the open bars in the bar graph on the left, and that is the unmodified ApoC3. And looking across the different subjects from 1 through 6, there's not a huge change in this proteoform between the 2 groups. The second group is identified by the squares in the upper right-hand side and the filled-in bars in the bar graph. This is the linear GalNAc-Gal sialic acid form on ApoC3. And in this proteoform, you can see a large change between the low HDL group and the high HDL group. And finally, the third group, identified by the triangles on the upper right and the hashed bars on the left bar graph, is this branched group, which showed very little change, again, between the low HDL group and the high HDL group. And this indicated that there was not only a change in ApoC3 between these 2 groups, but that it was specifically this triglyceride proteoform that was the main difference. So switching gears here to something that has been done in our group is that we study the process of senescence. And this is a process by which many cells in the body go through, where instead of proceeding through the cell cycle, they stop in the middle of G1 and do not proceed. And so these cells generally elongate and flatten and produce a variety of different markers, and are noted mostly by their possession of senescence-associated heterochromatin foci, or SAFs, where not a lot of DNA synthesis is going on, or in fact protein synthesis. So we wanted to look at proteins that were changed in this process. And so in the cell line that we looked at 3 different populations. First was a control, which exhibits a normal growth curve. The second is upon treatment with this chemotherapy agent. For 24 hours, these cells will undergo senescence. So we took a day 5 sample. And we also, in the same group, some of these proteins will actually escape from the senescent state. It's not well known why that happens. But we took that population as well at day 17. We wanted to look at differences in proteoforms between them. Okay, so the first protein that we look at is HMG-A1. These high mobility group proteins are responsible for chromatin architecture and the recruitment of different factors to turn on and off genes. The only difference between these 2 HMG-A1 forms is that HMG-A1A has a 10 or 11 amino acid group that A1B does not. And so we can look at changes now not only across the 3 different states we're looking, but even between these 2 proteoforms. So both of these proteoforms were well identified, but between the 2 proteoforms, the 2 different forms in the control group, we see that both of them exhibit high amounts of di, tri, and some tetraphosphorylation. But only in A1A do we see this methylation, which we have shown occurs on arginine 25. And A1B, while it contains that same arginine, we do not see any methylation occurring in any state. So now looking across the different treatments, we see that as A1A moves from control into a senescent state, we maintain our levels of phosphorylation, but now we have mono and even diphosphorylation occurring at that arginine residue, where we see very little change occurring in HMG A1B. And finally, in the escape population, we see that the proteoforms themselves do not return to the control population, but are somewhat different even still. And so the second protein I wanted to mention was HMG A2A, which is another high mobility group protein, where we were able to see in the senescent form 5 separate phosphorylations, and using top-down proteomics, we can isolate where each of these phosphorylations occurs. So the first 3 phosphorylations occur in the C-terminus of the protein. The 4th phosphorylation we found to occur on serine 44, and by isolating just the 5-phospho peak here, we find that that phosphorylation occurs at threonine 40. And so with that, I wanted to thank all of my collaborators at the Proteomics Center of Excellence at Northwestern University. We work with Professor Neil Kelleher. This is shown in the lower left, and the rest of my team is shown behind. Another thing I wanted to bring your attention to was that we have a portal for top-down proteomics established by the Consortium for Top-Down Proteomics, and you can get to that at http://www.topdownproteomics.org. And finally, I wanted to invite anyone to the Sanibel conference hosted by ASMS on top-down mass spectrometry that's happening in late January at St. Pete Beach in Florida. It's being hosted by Neil Kelleher and Liliana Pasatolich, and important deadlines, abstracts can be accepted through the 14th and registration through the 21st of December. And with that, I'll turn it back over to Tamlin.
Thank you very much, Paul. Before we move on to our next speaker, I want just to remind everyone that they can submit questions at any time by typing a question into the Ask a Question box on the lower left of the screen and hitting Submit. We'll answer as many questions as we can at the end of Pierre's presentation. Pierre, we're ready for you.
Thank you, Tamlin, and thank you for the opportunity to talk at this webinar. Thank you for all those that dialed in today. What I would like to talk about today are the recent analytical development in mass spectrometry that now facilitates the large-scale profiling of peptide antigen for cancer immunotherapy and also for personalized medicine. I will first describe the nature and origins of peptides bound to major histocompatibility complex that we collectively refer to as the immunopeptidome. I will then highlight the dynamic nature of the immunopeptidome and how the composition and abundance of the immunopeptidome are changing in response to environmental cues. And I will finish by describing how we can take advantage of the changes in the immunopeptidome between cancer and normal cells to develop an immunotherapeutic approach to eliminate neoplastic cells. Okay, let's first talk about an overview of the antigen presentation and processing machinery. From fish to human, nearly all cells in jaw vertebrate constitutively process and present peptide derived from exogenous and endogenous protein to cell surface molecule called major histocompatibility complex, or MHC for short. There are 2 classes of MHC molecule that are distinguishable from their structure and their intracellular pathways through which they encounter peptide antigen, and their recognition by different kinds of T cells. First, on the right, we have the MHC class 2 molecules that typically generated from a exogenous protein that are internalized into endosome and degraded by endosomal protease prior to being loaded on newly synthesized class II MHC molecules. This pathway is typically used by antigen-presenting cells such as dendritic cells, macrophage, and B lymphocytes. Now, the main focus of this talk will be on MHC class I, which is shown on the left. These are typically generated from protein degraded in the proteasome and the cytosol, and shortly after, they are transported into the lumen of the endoplasmic reticulum via the transporter associated with the antigen, also called TAP, and then loaded on newly synthesized MHC class I molecule before trafficking to the cell surface. This pathway operates constitutively in all O cells, so we see it in all the human and mammalian cells. The antigen that are presented are recognized by what we call CD8 T lymphocyte that are trained to elicit a immunological response if a cell displays the unusual antigen. In this case, they can actually kill the displaying cell or secrete cytokines such as interferon gamma or tumor necrosis factor, resulting in the local recruitment of other immunological effector. Probably what you are thinking about at the moment is what's so special about the immunopeptidome. As I indicated before, they represent peptide displayed by MHC molecules or antigen-presenting cells, and their selection is based on the specificity and affinity of the binding groove of the MHC molecule that we'll talk about shortly. Since they originate from O-cell protein, the MHC class I repertoire is highly diverse, and human cell can display more than about 5,000 different peptide per cell. They also vary widely in abundance, typically from 10 to 1,000 molecule per cell. As will be shown later, the abundance of these peptides is not correlated with protein or gene expression, but rather it is shaped by rapidly degraded polypeptide, including those that we commonly call as defective ribosomal product, that are processed via the proteasome or cytosolic and ER protease, as well as their affinity to MHC molecule, also called human leukocyte antigen in human. The MHC class I system also display important role in olfactory-based vertebrate mate selection in rodent, for example, and in tumor immunosurveillance and tissue rejection. What's so interesting about the MHC peptide is the fact that the MHC1 molecule, the receptor, will actually display a highly diverse population of peptide that range from 8 to 11 amino acid long. In laboratory mice, for example, we find predominantly the H2KB, H2DB, and QA2 allele that preferentially select peptide with specific motif, such as the one that I'm showing here at position 5 for leucine and position 9 for H2KB. In human, there's a wider distribution, as you can readily appreciate from the bottom portion of this slide. The most frequent allele that accounts for about 95% of Caucasian population are the 6 HLA displayed at the bottom of this slide. And as you can appreciate, there's a variety of different motifs that are present in these HLA molecules. The analysis of these peptides by mass spectrometry represent a significant analytical challenge, mainly because there are inherent diversity in peptide sequences and most importantly, the relatively low abundance. So for example, we typically need about 100 million to 1 billion cells to obtain sufficient amount of MHC peptide to detect them by mass spectrometry. To probe this diverse population of peptide, we also need a high-performance mass spectrometer that provides femtomal detection limit and also comprehensive identification via high-throughput MSMS sequencing. Now, one thing I should probably mention is that in contrast to tryptic peptide that has predictable structure with a C-terminus arginine or lysine residue that favors also the sequential cleavage of amide bond through peptide backbone, well, Mhc peptide don't necessarily generate evenly distributed intense fragment ion. So that's a major difficulty when identifying MHC peptide via MSMS. Also, another point is that Mhc peptide can contain polymorphic variant or even post-translational modification that renders their identification more complicated using conventional database searches. Now, to identify potential tumor-associated antigen, we also need to profile the abundance of Mhc peptide across different sample sets and then subsequently validate them through immunological response with T-cell assays. Over the past 20 years or so, different approaches have been used to isolate Mhc peptide. The first approach that was pioneered by the group of Don Hunt in the early 1990s used antibodies that recognize specific HLA molecules. And peptides were then released from the immunopurified MHC complex using acid buffer and then filtered on a 5 kilodalton cutoff membrane prior to mass spec analysis. The second approach that is highlighted in the mid portion of this slide uses cell lines that express soluble HLA molecules that are not retained at the plasma membrane and are secreted outside of the cell, which are very convenient for isolation. The MHC complex that is isolated by immunoaffinity purification, and peptides that are released by acid dilution, and then they're filtered on a membrane and analyzed by mass spectrometry. The last approach that is on the right and is also the one that we're commonly using is more generic. It does not involve any antibody purification and simply use bile acid dilution with citrate buffer to release peptide from their MHC complex. In this case, we obviously need to ensure that cells are viable through this process and that we minimize cell lysis. Peptide that we obtain are purified on a low molecular weight cutoff membrane, purified on HLB cartridges before mass spec analysis. Okay, now if we turn to the identification of Mhc peptide, in our particular case, for example, if we examine control and neoplastic lymphoma EL4 cell from mouse, we also compare the peptide distribution against a B2 microglobulin knockout that is not expressing MHC molecules, so that we specifically determine peptides that are truly associated with the MHC molecule. All samples that we've analyzed are analyzed on a LTQ Orbitrap instrument that provides high sensitivity and throughput for peptide sequencing. Typically, we would be acquiring a high resolution mass spectrum to obtain accurate peptide mass measurement in the Orbitrap analyzer, and then we follow that up with 12 MSMS scan in the ion trap, so that gives us a duty cycle of just under 2 seconds. So from our MSMS spectra, we search them against a concatenated forward and reverse mouse protein database to identify peptide with a false discovery rate of less than 5%. And we also align all the LCMS peptide maps with a tolerance of 5 ppm on mass and plus or minus 1 minute on time, and then correlate the peptide identification across sample sets to identify differentially abundant peptide, as highlighted on this panel. For example, by comparing peptide abundance from wild type and beta-2 knockout, we can differentiate contaminant peptides from genuine MHC-1 peptide. We also filter out peptide candidate to only retain those that have the required 8 to 11 amino acid length with the expected binding motif. For example, when we compared in a previous study the peptides from wild type and beta-2 knockout, we identified about 181 peptides that were specifically reproducibly detected in the wild type that represented about a third of the entire peptide population. This is shown in the volcano plot on the left that shows the log2 fold change of abundance ratio between the wild type and the knockout EL4 cell versus the log10 p-value from triplicate measurement of the corresponding samples. Now, out of these peptides, around 200 had a consensus motif for H2DB, H2KB, and QA1 or QA2 MH1 molecule, and more than about 80% of these identifications were found to be on the top 1% of the IS binder based on SMM prediction algorithm. Now, an interesting finding that we report at that time is the fact that we uncovered that the immunopeptidome repertoire from the thymocyte conceals a tissue-specific signature that derives from about 17% of MHC1-associated peptide. This is shown on this slide where we rank our peptide source gene based on mean expression value for 36,000 transcripts for each particular tissue available from the SEMATLAS database. We also developed a Z-score that we use to calculate for each peptide source gene to identify transcripts that were preferentially expressed in the thymus. Now, if we display this on a heat map that shows the relative mRNA expression for 58 tissue of the 30 peptide source gene with the IS thymic Z-score, we can see that the IS match were obtained for tissue representing bone marrow, lymph node, and thymus, and the top ranking source gene are shown on the right. Another interesting finding that we reported recently is the fact that, like the proteome, the immunopeptidome of antigen-presenting cell is also very dynamic. This is shown in here in a recent paper that we published in Molecular System Biology, where we look at the dynamic change of Mhc peptide from thymic cell in response to rapamycin, which is an important inhibitor of the serine-threonine kinase mTOR that specifically regulates cell size and protein synthesis. In this study, we took snapshots of the immunopeptidome in control and lymphoma cells exposed to rapamycin for 0, 6, 12, 24, and 48 hours. And the first thing we saw is an overall increase in the abundance of the Mhc peptide compared to control cells for each allele, as shown in the volcano plot on the right. This is also illustrated on the next slide here, where on the left panel we identify specific source proteins such as RICTOR, the rapamycin-insensitive companion of mTOR, and CDK activating kinase assembly factor MAT1. What we can see here at the bottom right scatter plot is the protein level for these two proteins that remain relatively constant over the 48 hours, but the MHC peptide derived from these proteins increased either at 12 hours for MAT1 or 48 hours for RICTOR. So what we can see from these data is the fact that the immunopeptidome is dynamic and does not necessarily correlate with the expression or the stability of the native form of source protein. Now, one question that is probably very central to what we do is that we wanted to answer whether or not the immunopeptidome peptide that are differentially abundant in control and cancer cell can elicit a proper immunological response and can be used as a vaccine against neoplastic cells. Now, to address this question, what we've done is we compared the immunopeptidome of control thymocyte against the neoplastic thymocytes from mice injected with the EL4 lymphocyte cells. Now, the volcano plot representation that you see at the bottom left illustrate the MHC peptide that are reproducibly detected across biological triplicate. and I highlighted two peptides from CDK inhibitor 1B and also X-link regulated protein 3 that were significantly down in ROP regulated in control and no plastic phagocyte cells. Now, as an example, the ion chromatogram of the doubly charged ion at 471 from Van Reuvel peptide is shown at the bottom right with its corresponding MS-MS spectrum confirming the sequence assignment. Now, these analyses identify about 200 Mhc peptide, of which approximately 10% were differentially regulated. And an example of that is shown in the next slide. So these are tumor-associated candidates that represent a subset of the differentially abundant peptide with upregulated peptide in red and downregulated peptide in green. Now, to test if the differentially regulated peptide can be used as vaccine for immunotherapy, we selected two candidates that I've highlighted with the arrows on this slide. They represent the XLR3 and SJK, and we used them to prime T cells. And how we did that was by loading the selected peptide on dendritic cells that we subsequently expose to naive T cells so that they can specifically recognize these antigens. So the corresponding activated cytotoxic T cells are expanded in the recipient mice and thus are trained to recognize the corresponding tumor-associated antigen and can lyse and kill the tumor cell that harbor these antigen. Now, the key piece of information is shown on this slide, which highlights the survival curve of unvaccinated and vaccinated mice. In each case, we use 10 mice per group. And as you can see, the unvaccinated mice that were injected with no plastic AL4 cells did not survive more than 35 days, whereas the mice immunized with dendritic cells that harbor either of the two peptide candidates were competent to eradicate the neoplastic cell. So these data essentially confirm that differentially abundant peptide from the immunopeptidome and of neoplastic cells can be used for immunotherapy. Now, I'd like to maybe finish on an interesting development that we recently made in expanding our immunopeptidomic study to investigate its application to human cancer, and more specifically leukemia. As you probably know, leukemia is the fifth most frequent cancer, and the current treatment is allogeneic hematopoietic stem cell transplantation to prevent the bone marrow toxicity that is caused by intensive chemoradiotherapy regimen. Now, however, harnessing the graft versus leukemia or GVL effect to improve the outcome for patients with leukemia has proven to be quite challenging because about two-thirds of the patient will develop an autoimmune response that is commonly referred to as graft versus host disease or GVHD that shows up as that manifests itself in the skin, gastrointestinal tract, and or liver. And an example of that is shown for a patient here, 42-year-old patient that suffer from GVHD. Now, what we want to develop, obviously, is another more effective immunotherapy treatment that is based on minor antigen. This will be obviously a key component of our immunotherapy treatment to specifically eradicate leukemic cell. Now, minor antigen are a subset of the Mhc peptide, and they represent polymorphic peptide that are displayed also by the HLA molecule of recipient cell. Now, T cells that recognize the minor antigen that are only expressed by recipient hematopoietic cell, including leukemic cell, could contribute to the specific elimination of leukemic cell without having as a side reaction the GVHD. The ideal minor antigen would be derived from proteins that are not only selectively expressed in hematopoietic cell, including leukemia cell, but also necessary for their function. So that obviously would minimize the potential for leukemia cells to evade the T cell recognition by losing the expression of the antigen. So you may wonder, what are minor antigen? So as I mentioned before, the minor antigens are peptides from source protein that contain single-nucleotide polymorphism, also known as SNP, as Paul alluded to before, and they result in slightly different expressed product. Now, in MHC matched transplant, when the antigen-presenting cell do not display a peptide variant, the recognition by a donor T cell does not elicit a directed activation of T cells, and antigen present in the cells remain intact. Now, this is not the case for MHC mismatch and transplant, where the MHC molecule will lead to increased binding and activation of T cells to antigen-presenting cell, eventually leading to the targeted elimination of leukemic cell that specifically harbor the corresponding minor antigen. From a mass spectrometry point of view, there's two types of SNPs. There's a synonymous SNP that cannot be distinguished from the reference counterpart, as the peptide sequences are identical and not readily matched in the reference database. However, a non-synonymous SNP that is not present in the reference database will not be matched by the search engine if we use the reference database. Now, the consequence of non-synonymous SNP on the immunopeptidome are twofold. First, a SNP within the Mhc peptide can lead to unassigned MSMS spectrum unique to a specific patient. And the second aspect is if the SNP is located in the flanking region, this can also affect the processing and presentation of the Mhc peptide, and that can result in a match MSMS spectrum for which there is a differential abundance. There is a differential abundance of the corresponding peptide. Over the past 15 years or so, there's no more than 50 minor antigens that have been identified in human, reflecting the difficulty in the large-scale identification of these important antigens. So that prompted us to look into ways to identify the minor antigen. But they're obviously very challenging analyte to identify because SNP obviously occur in a low frequency along the 3 billion base pair in human genome. And we estimate that that minor antigen represent about a subset of less than 1% of the entire MHC peptide population. And that highlights the fact that we need very comprehensive MSMS sampling to get at the minor change that we can expect from the immunopeptidome. Since minor antigens are also occurring at low copy number per cell, then the MSMS requires very high sensitivity instrument. As I mentioned before, the identification of polymorphic variants require that we either perform de novo peptide sequencing from the unassigned MSMS spectra, or that we conduct search on individualized protein database for each patient. In either case, we need high-resolution MSMS, and that is certainly beneficial for this type of identification. Eventually, to confirm the peptide identity and validate the immunogenicity across a wide array of individuals, we need to do follow-up experiment that can be done with transcriptomic bioinformatics to evaluate the frequency of the variant and also T-cell assays. Now, this slide shows the minor antigen discovery platform that we developed. In a preliminary set of experiment, we've used EBV transform B lymphoblastoid cells line from peripheral blood mononuclear cells of HLA-matched individuals. We isolated the Mhc peptide using mild acid dilution, as I described before, and we analyzed the sample in biological triplicate by LC-MS on a Orbitrap Elite instrument. We also obtained individualized database from the DNA and RNA sequencing using a Illumina iSeq instrument. And we also evaluated the potential of de novo sequencing software, such as Peaks, to identify potential minor antigen. Our list of minor antigen, once we identified these by MSMS, is further refined by applying criteria such as peptide length, HLA binding affinity, and allele distribution. Here's an overview of the MHC1 peptide that we identified in this preliminary experiment on HLA-matched sibling. So from more than about 26,000 MSMS that were assigned, we identified approximately 3,400 unique MHC1 peptide by using conservative filter criteria on peptide length, false discovery rate, and HLA binding affinity. Approximately 96% of these peptides were common to both siblings, while a subset of 86 peptides that I highlighted in red and green were differentially abundant in one of the two patient B cells. An example of a peptide showing a five-fold increase in patient M compared to patient R is highlighted by the arrow here, and it corresponds to TNF-alpha-induced protein 8, like protein 1, or TIP1 for short, which is a member of the TNF-AIP8 family that is important for the maintenance of immune hemostasis. There's a recent evidence that suggests that TIP1 may perform function in cell secretion and carcinogenesis. Interestingly, we also noted that out of the 3,400 Mhc peptide identified, there's approximately 1% that were detected in one patient only. An example of this is shown here for the case of ZW10 Intractor, a known component of the kinetochore complex required for mitotic spindle checkpoint. Interestingly, this peptide was matched to the reference database and was only detected in patient R, but a variant form where the arginine was substituted by glycine was found in both patients. This variant is also known as SNP, but we further confirmed it with using Sanger sequencing, as shown on the right panel here. Another example of a minor antigen that is entirely new is the case for a peptide that is associated with a uncharacterized protein that we identified unique to patient R, and that corresponds to a substitution of the C-terminal glycine for an arginine. Now, one thing that is very interesting about this substitution is it confers a significantly higher binding affinity to HLA A0301, because the arginine is typically well represented in the C-terminus anchor site of the allele motif. Now, this table presents a couple of examples of identifying minor antigen that we correlated with genomic sequencing. And interestingly, what we note is that there's in a number of cases, the SNP is localized at a different position than the anchor site, and this results in limited impact of the predicted affinity, and the corresponding peptide is typically observed in our immunopeptidome dataset. However, in contrast to this, when the substitution takes place at the anchor site for a given allele, the peptide can exhibit a significantly lower affinity and may not be observed in the immunopeptidome of the patient. And this is the case for the peptide that I showed in the previous slide. Now, in summary, what I'd like to leave for the audience is the fact that the recent advance in mass spectrometry has provided enhanced sensitivity, resolution, and throughput that now facilitate the large-scale sequencing of Mhc peptide. And of particular interest is the ability to perform de novo sequencing or database search with a personalized database to identify minor antigen that can be used for immunotherapy. Obviously, sensitivity and comprehensiveness of MS instruments such as the Orbitrap Elite enables the identification of low abundance minor antigen that represent less than 1% of the entire Mhc peptide population. Now, interestingly, these advances open up new perspective in immunotherapy for more effective graft versus leukemia effect, and obviously its application, broader application to the development of T-cell based immunotherapy. Now, I'd like to finish by acknowledging the contribution of different lab members, and in particular Dev Sriranganadan and Olivier Caron-Lizotte that contributed directly to this work, but also my collaborator from the group of Claude Perrault, also at ERIC, and also his student and postdoc that contributed to this project. And finally, our funding agency for financial support through this project. So this concludes this part, and now we're back to Tamlin.
Thanks, Pierre. All right, we're ready to start our Q&A discussion. We have a lot of good questions, and we'll answer as many as we can. You still have time to submit questions, though, so if you have any, send them in right away. Okay, Paul, our first question is for you. What sorts of projects are best suited to top-down proteomics?
Well, some of the better projects for top-down proteomics are when you're looking at proteins that are smaller and more abundant. Top-down proteomics still has some limitations in terms of its upper mass range, but with small proteins below, say, 40 kilodaltons or so, you can really get some spectacular results that'll give you more information than you would expect with bottom-up.
All right, Pierre, the next one's for you. Why can't we determine minor antigens from the genomics database containing polymorphic variants?
Well, it's possible to predict potential minor antigen based on SNP database and HLA motif analysis. However, the mechanism by which the Mhc peptide are generated and selected are less defined, and we cannot predict it from the human genome per se. Also, we cannot predict the relative abundance of the corresponding peptide at the cell surface, so our only option currently is to perform mass spec analysis to define the composition and nature of the immunopeptidome and the minor antigen.
All right, Pierre, this another one for you. Is the abundance of SNPs and MHCI significant enough to cause transplant tissue rejection?
Yeah, so it's known, for example, that a T cell can certainly recognize even 1 to 10 copies or molecule of the Mhc peptide at the cell surface. So if the T cell are properly trained to recognize the corresponding minor antigen, then obviously they can elicit the proper immune response.
All right, Pierre, I do have a bunch of questions for you. Here's another one. So then, for cancer treatment, this process has to be done for each patient. Do you expect someday to identify an immunopeptide that can be used on rare subsets of patients for specific cancer types?
Okay, so obviously this is the basis for the personalized medicine that is based on Mhc peptide or immunopeptidome. So we expect that one day with the improvement in technology, we can certainly profile the immunopeptidome of each patient so we can provide a accurate account of the distribution of Mhc peptide to better define, for example, which would be the best minor antigen for immunotherapy. Also, the we can use tumor-associated antigen. They're not obviously minor antigen, but with the caveat, obviously, that this might not be a directed approach to specifically target, for example, leukemic cell. And this is the reason why minor antigen are better antigen than the tumor-associated antigen to prevent any autoimmune disease or GVHD in the case of leukemia.
Okay, Paul, we're over to you now. Any examples of top-down used for rare ubiquitin linkages and branching?
Well, top-down could be used for that, but the problem comes in that the modification, ubiquitination or simulation or modifications like that are quite large. They're 8 kilodaltons themselves. So you really start to reach the upper mass limit. And really, monoubiquitination is about what one would expect to see in top-down. Once you get to the branching and polyubiquitination, it becomes much more difficult.
All right, another one for you, Paul. Can you elaborate how to do de novo amino acid sequencing using MS-MS?
Sure, it's a very long answer if you wanted to go through exactly how to do it. But it's typically, you take a peptide or a small protein and you fragment it. You can look at the fragments that are observed, determine what their neutral masses are from the charged masses that you see, and the spacing between them, if you have a really good mass spectrometer and good fragmentation methods, should be that of a single amino acid residue or maybe two, and start at one end of the protein or maybe somewhere in the middle and start building up your sequence.
All right, Pierre, we're back to you. Is the immunopeptidome enriched in polymorphic variants?
Well, the short answer is probably not. Our preliminary data indicates that the proportion of minor antigen is a bit less than about 1%, and this is consistent with what is predicted from the proportion of nonsense SNP deduced from the genomic sequencing. So I would say at the moment we think that it's probably not enriched.
Okay, and I have another question for you. Why are MHC-1 peptides more difficult to identify by MS-MS compared to tryptic peptides commonly sequenced in bottom-up proteomics?
Okay, well, there are several reasons associated with this difficulty. The first one is that Mhc peptide do not have fixed or predictable motif in contrast to tryptic peptide that have lysine or arginine residue at the C-terminus and at the N-terminus of the flanking amino acid. This variability in peptide sequence dictate that our database search use no defined enzymatic restriction. And this increases, obviously, the number of potential sequence candidates. The second reason is that the fragmentation of Mhc peptide do not necessarily generate evenly distributed B and Y fragmenti that are typically used for sequencing peptide. And these ions obviously are more typically observed in tryptic peptide. The consequence, obviously, of these effect is that for the same sort of false discovery rate, we obtain significantly higher mascot score requirement for MHC peptide compared to tryptic peptide. And obviously, this is a major bottleneck in our database searching.
All right, Paul, we're back to you. How are PTMs searched in top-down proteomics, and how does that compare to traditional bottom-up proteomics?
Oh, sure. It follows on nicely with Pierre's question. It's a more complex search space. So traditional combinatorial PTM expansion can't really be done in top-down proteomics. We can't predict any potential PTMs. What we do is we rely on what is in the literature. So using the UniProt database, we're able to determine what has been seen previously, and we look for those. And we can also look for certain PTMs that are unknown based on differences in intact mass between the observed and the theoretical forms.
Okay, thanks, Paul. Unfortunately, our time is up. Thanks for listening in, and thanks for all the great questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, Thermo Scientific. We'll be sending you a survey shortly and hope you take the time to respond. Your comments help us provide topical and timely webinars. Thank you.